we are planning to discuss is about retina. Okay, and we already know, okay, what is retina. It is the sensory layer at the back surface or back side of the eye. Okay, as we just remember, the eyeball has three different coats. The one that is the outside fibrous coat, and then you have your choroid, ciliary body, and iris as the as the vascular coat, and inside you have the retina as the neural coat. So if you see that this is the whole is retina, it is not only one part. Do not confuse with this retina because it's actually vitreous humor, but to show the whole surface of the retina, they have put the retina uh, thing over here. So the retina is starting from this point. This is the ora serrata all across ora serrata. This is also ora serrata, and it is extending towards the optic nerve head. Again, optic nerve head is a point where all the nerve fibers from all over the retina will get collected, and it is passing through the optic nerve to the brain for further processing. So if you remember the light rays that is passing through the cornea, passing through the pupil, through the crystalline lens, and through the transparent vitreous, and getting hit on, and retina is the layer that is transferring the light. So here are the learning objectives. Okay, we are going to learn the basic anatomy of the retina, and it has multiple layers, in fact 10 layers. Okay, we will be talking about different layers of the retina. Okay, all the layers of the retina we need to remember, as well as we need to learn, okay, each one of separately. And we also will be describing the functions and features of each retinal layer, and we also have to learn different regions of retina. Okay, for example, this is a central part of the retina, and this is a peripheral part of the retina. So central retina, this is a central retina. Okay, peripheral retina, as well as we need to also talk about the optic disc. Along with that, we have to also talk about macula, fovea, and Phobiola. And towards the end we'll be discussing about the blood supply of the retina. Okay, how the blood vessels are entering to retina, as well as how it is spreading all over the retina, and what are the different names of each parts of the blood vessels. So as we discussed in the first slide, retina, we already understood that it is the innermost coat of the eye. Okay, this is just a revision what we discussed in the first slide. It is called the neural layer, located between the choroid and the vitreous. So we already know that this whole area will be filled by vitreous, and it is this just the brown one above that retina is the choroid, and the white one, as we already know, that is the sclera. So it is between the choroid and the vitreous, and as we mentioned, it extends, retina extends from the optic disc to the ora serrata. This is the optic disc, and it is extending to the ora serrata, optic disc, and to the ora serrata. And the third point also we discussed already, it transfers the light energy into neural signals so that the neurons can easily pass through the neural layers or through the optic nerve and into the brain for further processing. And it contains first three cells of the visual pathway. So we'll be discussing the visual pathway a little bit in detail later. Okay, retina is the first part of the visual pathway, and then optic nerve, and it is going through the chiasma, going to the lateral geniculate body to the brain. But do not focus on that now. We'll talk about retina. So as I mentioned, visual pathway, the first part of the visual pathway is the retina. So it contains the first three cells in visual pathway. In fact, all these three cells are part of the retinal layers. That means the photoreceptor cells, bipolar cells, and ganglion cell. Do not worry about that now. In the subsequent slides, we'll be learning about each of these. Okay, what are photoreceptor cells, what are bipolar cells, and what are ganglion cells. Okay, here you go. Okay, this is how a bigger picture of the retina, how it will look like. Okay, you can see that this is the picture of the eye. The light is coming through the cornea, through the pupil, through the lens, and through the vitreous onto the retina. And if you look at this picture, this is a bigger magnified picture of the retina. Okay, and if you want to understand, your choroid is here, right? This is the choroid, and your vitreous is here. Okay, so this whole thickness is retina, and as I mentioned, you can see so many different layers of cells. Okay, and that is what exactly the different layers of retina means. Okay, so now one interesting thing you all will be thinking that, okay, when the light is passing through the vitreous and coming and hitting onto the retina, from the retina the light is passing through the different layers, okay, and the final processing before it reaches, or sorry, before it goes out of the eye, will be happening at the back surface. It is not. So what actually happens is, when the light come and hit the retina, it pass through all the layers of retina and go to the back or the posterior most layer of the retina. From there the light processing starts, and this light will again travel through the different layers of the retina. Okay, so first it will go and hit the second layer of the retina. When we learn different layers, you will understand that. So do not think that the processing starts from the anterior side of the retina. When the light enter, it passes through the layers of the retina and reach the posterior most layer of the retina, and from the posterior most layer the processing of the light starts. And once that processing starts, light will again pass through different layers, and then it will become the neural signals at the anterior side, and these neural signals will be passing through, this is the optic nerve, and outside the eye. So this is an important thing which all of us have to remember. Okay, this is the vitreous side, this is the choroid side, the whole thing is retina. When the light enter, it passes through the different layers of retina without getting processed, and goes to the posterior most layer of the retina, where you have the photoreceptor cells. That photoreceptor cells will receive the light rays and then start processing the light rays. So you can see this blue color and orange color, all these are photoreceptor cells. The meaning of photoreceptor cells is nothing but photo is light, receptor is that receives. So the light that is entering the eye passes through all the layers of the retina, reaches to the back side of the retina, that is the posterior most layer of the retina, and there you have the photoreceptor cells, and that photoreceptor cells will receive the light, start processing the light, and then start sending through the cells of the retina, and by the time it reaches here, it becomes the neural signals. And so these neural signals will get collected by the optic nerve, and it will go into the through the optic nerve to the brain for further processing. I hope you understand. Okay, here we go. Okay, these are the 10 different layers of the retina. Okay, as I mentioned, it is starting from posterior to anterior. From the previous picture, this is the posterior side, that is the choroid side, and this is the vitreous side, that is the anterior most side or anterior most layer of the retina. So these layers are arranged from posterior to anterior. So the first layer is retinal pigment epithelium. You can see here this dotted one, that is the retinal pigment epithelium, so that is the posterior most layer. The second layer is the photoreceptor cells. I just discussed about this in the previous slide. Photoreceptors, okay, that is receiving the light, and this photoreceptor cells has different parts, and which we'll be discussing in the subsequent slides. So this external limiting membrane is also a part of the photoreceptor layer. Outer nuclear layer, that means this layer has nucleuses. We know that every single cells in our body has a nucleus, and this is the nuclear layer. You can see, if you look at here, there are small dots here, okay, nucleuses for each of these photoreceptor cells. So the layer of a set of this nucleus cell layer, so this level is the outer nuclear layer, and you can see that photoreceptors are coming and connecting to another set of cells, so that is the outer plexiform layer. Plexiform is nothing but getting connected, and then comes the another layer of nucleus. As you can see, this dots over here, that is the nucleuses of the next layer. And then there is one more connection junction. These cells are getting connected to the final set of cells, so that is the inner nuclear and inner plexiform layer. And you can see this purple color is the ganglion cell layer, and from the ganglion cell layer the nerve fiber layer and the internal limiting membrane. So we will understand this one more clearer, okay, in the next slide. So what you need to remember is, okay, the retina has three types of cells: photoreceptor cells, okay, and these are the bipolar cells, and these are the ganglion cells. So photoreceptor, bipolar, and ganglion cells. So those are three different sets of cells in the retina, and other 10 layers, all these 10 layers are at least, or they are either a part of any of these cellular components. Okay, so we'll be able to explain a bit more clearer about each of these layers in the next slide. Okay, here we go. Here we have a bit more broader picture of the retinal layers. Okay, so now we have to slightly readjust yourself. Okay, here is a choroid, this is the posterior, and here is a vitreous, that is the anterior part. Okay, so this whole distance is the retina. Okay, so this will be just a revision of what we discussed, the points in the previous slide. So these are the 10 layers starting from the retinal pigment epithelium. So whatever you are seeing here, okay, this one is retinal pigment epithelium, and okay, your photoreceptor cells are this much. These are the photoreceptor cells, and these cells are the bipolar cells, okay, bipolar cells, and these cells are the ganglion cells. So you need to remember that there are three types of cells. One is photoreceptor cells, second is bipolar cells, third one is ganglion cells. Now we'll come to different layers. Okay, so remember that all these layers are part of these three sets of cells. Now retinal pigment epithelium, we'll talk about retinal pigment epithelium, or RPE, in detail in the next slide. So the photoreceptor cells are connected to the retinal pigment epithelium. Okay, and that is a basic function of retinal pigment epithelium, which we will learn in the next slide. So retinal pigment epithelium, then your second layer is the photoreceptor cell. In fact, this the whole thing is photoreceptors. Okay, so photoreceptor layer, and the next layer is external limiting membrane. Okay, so the photoreceptor cell has an outer segment and it has an inner segment. We will be learning about photoreceptor cells also in detail, so at that time we will understand. So photoreceptor cells has an outer segment and it has an inner segment. So the junction between the junction between the inner segment and outer segment of the photoreceptor cells is the external limiting membrane. Okay, so retinal pigment epithelium, photoreceptor cells, external limiting membrane. So what is external limiting membrane? It is junction between the inner segment and outer segment of photoreceptor cells. Now the next layer after external limiting is the outer nuclear layer. Now look at the cells here. Okay, within each cells there you can see some dots. Those are the nucleus of these cells. See, every cell in our body has a nucleus. So this layer, okay, where the nucleuses of photoreceptor cells are known as the outer nuclear layer. Okay, now the next one is outer plexiform layer. Now what is the meaning of plexiform? Plexiform means junctions. Okay, so you can see that the photoreceptor cells are connecting to bipolar cells. You understand? So the junction between the photoreceptor cells and bipolar cells, this junction is known as outer plexiform layer. So remember that plexiform means connecting. Now after the outer plexiform, when you come here, you can see that it is becoming inner nuclear layer. Again, you can see these dots in the bipolar cells. So that means the nucleus of bipolar cells, that is known as inner nuclear layer. So after the nuclear, again you can see that another plexiform comes, nothing but junction between the bipolar cells and ganglion cell. That is the inner plexiform layer. Okay, so inner plexiform, then of course your ganglion cells. Okay, there is no nuclear layer of ganglion cell as a separate one. This is a ganglion cell layer. Okay, and this ganglion cell layer, okay, is transferring as nerve fiber layer. So the light energy which is starting from here as light, through the processing between these cells, will become neural signals by the time it reaches the ganglion cells, and these neural signals are, okay, transferred into the optic nerve and going out of the eye. Okay, so there is one easier way to remember this. Okay, you can see that here it is outer nuclear, outer plexiform. Okay, so outer nuclear because photoreceptor cell is the outermost, that is why outer nuclear, and ganglion cells are the inner one, that is why inner nuclear, inner plexiform, and then forming the ganglion cells and nerve fiber layer. And the last one is the internal limiting membrane, that is a membrane that is between the vitreous and the ganglion cells. Okay, so you have one internal limiting, you have one external limiting also. So it is easy to remember. The outermost is retinal pigment epithelium, then to photoreceptor cells, external limiting membrane, that is the separation between the inner segment and outer segment of photoreceptor cells. And then the outer nuclear layer, it is nothing but the nucleuses of photoreceptors. Outer plexiform is the junction between the photoreceptor and bipolar cells. Inner nuclear is a nuclear layer of your bipolar cells. And again one more junction comes, the plexiform layer between the bipolar cells and the ganglion cells. And again after that it is a ganglion cell layer, and by the time the light reaches the ganglion cell layer, it has already been converted into neural signals. It will be collected by the nerve fiber layer and will be taken out of the eye. And there is a small layer, that is internal limiting membrane, okay, that is separating vitreous from the retina. Right, so in this slide we'll be discussing about the first layer, that is retinal pigment epithelium. So as we remember now, the retinal pigment epithelium is the posterior most layer which is situating closer to the vitreous. So in this picture, closer to the choroid, this is the choroid position and this is the vitreous position, and you can see that these are retinal pigment epithelium. Okay, and into the retinal pigment epithelium is your photoreceptor cells are connected. So it's interesting. So how many photoreceptor cells are there? There are near to 4 to 6 million retinal pigment epithelial cells are there into or in our retina. So you remember how much is this number? You can just imagine in Oman we have 5 million people, okay, including the residents and citizens. We have only 5 million people. Each of our eye, okay, you take your right eye, okay, it has around 4 to 6 million retinal pigment epithelial cells, so it is numerous, right? And each cell connects up to 30 to 40 photoreceptors. So this is just a photographic representation. Photoreceptors are connected to the retinal pigment epithelium. So each of the retinal pigment epithelium is connected to 30 to 40 photoreceptors. So now if you want to find out how many million photoreceptors are there, you have to just multiply say 40 into say 5 or 6 million. So that many millions of photoreceptor cells are there in the retina. So the retina is a very complicated, very sophisticated structure that is processing the light for us. right? So retinal pigment epithelium, okay, now we remember 4 to 6 million, and each retinal pigment epithelium is connected to 30 to 40 photoreceptors. Okay, another important thing is it has the retinal pigment epithelium, or RPE, has two types of pigments. One is called melanin, another is lipofuscin. So remember the spelling of these two terminologies, which is new for you. You might have heard about melanin. Melanin is a pigment that is giving color to the skin. So melanin is seen in the skin, it is seen in the iris. You remember iris has brown color. Okay, it is because of the melanin pigmentation, and also melanin pigmentation is there in the retinal pigment epithelium. So because of these two pigments, okay, these cells has got the name retinal pigment epithelium. Now these cells are highly metabolically active. Now you remember what is metabolism, right? We discussed metabolism in cornea. Corneal epithelial cells are metabolically active. We discussed metabolism in crystalline lens. The crystalline lens epithelium just underneath the capsule are highly metabolically active. So epithelial cells are having the function of metabolism, and metabolism is nothing but making energy. So, and you can now imagine why the photoreceptor cells are connected to the retinal pigment epithelium, because these photoreceptor cells are not having any blood vessels. So these photoreceptor cells are getting energy, okay, for its functioning. I told that whatever the light is coming to the photoreceptor cells are getting processed in the photoreceptor cells. So who is giving energy for this processing? Okay, your your retinal pigment epithelium is giving energy for this processing. That is why RPE, or retinal pigment epithelium, is highly metabolic. And now you can easily remember why, from where this retinal pigment epithelium is getting uh, oxygen and nutrients, because you have your choroid sitting here, and choroid has a very rich amount of retinal blood vessels. So those blood vessels are supplying, okay, the retinal pigment epithelium all the nutrients and oxygen, and it is giving that energy for this photoreceptor cells for further processing of light. It's interesting, right? So these epithelial cells are highly metabolically active, okay, because they are giving energy to photoreceptor cells and receiving blood from the choroidal blood vessels, okay, so that it can always and continuously receive the oxygen and nutrients. Now it will be very easy for you to understand why healthy retinal pigment epithelium is vital for the survival of photoreceptor cells. The meaning of vital is nothing but important and crucial. Survival is just for the living, right? Okay, so the meaning, if you are not survived, okay, if the retinal pigment epithelium is not supplying energy, your photoreceptor cells will die. So it is very important that the retinal pigment epithelium is healthy, so that the photoreceptor cells can receive energy and function. Okay, so there are some diseases, okay, that is happening in the old age, where the retinal pigment epithelial cells are getting damaged. So you can now imagine, if RPE, or retinal pigment epithelium, is damaged, it will affect the functioning of photoreceptor cells. And if RPE is damaged, photoreceptor cells cannot function. Okay, the light rays coming into the eye will not be transferred as neural signals and sent into the brain. So RPE, or retinal pigment epithelium, is very important. Okay, here we go. These are the functions of retinal pigment epithelium. So we already understood what is the basic function of retinal pigment epithelium, isn't it? Because it is taking care of the photoreceptor cells. So what are the functions? It controls the movement of nutrients from the choroid to the retina. Okay, so uh, retinal pigment epithelium is the one that is controlling the movement of nutrients. Okay, we already understood that nutrients are available, oxygen is available in the choroid. So from choroid to the retina, your artery is controlling. Second function, phagocytosis. Now, phagocytosis is nothing but, okay, it eats. Okay, the retinal pigment epithelium eats and clears okay, the cell components from photoreceptor cells. Now you remember that photoreceptor cell outer segment is the area where the processing of light rays happen. So during that process, a lot of waste products will be formed. These waste products from the photoreceptor outer segment is eaten by or is cleaned by the retinal pigment epithelium. So you can see that two basic functions. One is supplying nutrients for the retina and photoreceptor cells, and second one, it cleans or phagocytosis or eats, okay, the fragments of photoreceptor cells when uh, so all these waste products that happens during the process of transferring light into neural signals. So all those waste products are cleaned by the retinal pigment epithelium. It has one more function. Another thing is it metabolizes and stores vitamin A. Now we'll be discussing about vitamin A later, because vitamin A is a very important vitamin that is important for the processing of lights or light energy into the neural energy. Okay, so when we are discussing the visual cycle, we'll be able to understand it better. So it metabolizes, that means it produces vitamin A and stores vitamin A. Okay, and the last one is we already know that retinal pigment epithelium. There are pigments in the retina. Those are your lipofuscin and melanin pigments. Those pigments absorb light. So even if we have so much of light coming inside the eye, we will not be having so much of disturbance. These scattered light, okay, the light rays that are not perfectly focused or scattered light is absorbed by the pigments of your melanin, and lipofuscin pigments will absorb, so that our vision will be much more comfortable. So four basic function. One is to control the nutrients, the movement of nutrients from choroid to retina. Is the or RP is the intermediary, and it eats out or cleans out the cellular waste materials from the photoreceptor cells. It produces and stores vitamin A. Also, the pigments in the retinal pigment epithelium absorb light. So even if we have so much of bright light coming into the eye, our eye will be comfortable to accept that. Okay, we are coming into the second layer, that is the photoreceptor cells. Okay, so we already completed the first layer, retinal pigment epithelium or RP. This is a photoreceptor cell, that is the second layer. So these photoreceptors, from as the name suggests, these are the specialized cells in the retina that can receive light. That is the name, photoreceptor. Photo stands for light, receptor stands for receiving capacity. So there are two types of photoreceptor cells. Okay, one is rods, another one is cones. You may be remembering, okay, why rods and cones. Okay, these cells are named based on their shapes. So I have given an example. Okay, you can see that this has a shape of a cone and this has a shape of a cylinder or a rod. So the cells that has shape of a cylinder, okay, now you can see that this cone shaped, okay, it's generally the shape of the outer segment. Okay, so this is the outer segment of a rod and outer segment of a cone, and also even the inner segment has the similar shape. So why the name? Okay, it is because of the shape of the cells. So you have rods and cones, and this rods and cones also has specific pigments. So we learned that in retinal pigment epithelium there are two types of pigments. One is melanin, another one is lipofuscin. But in photoreceptor cells, each of the photoreceptor cells has specific pigments in it. Okay, rods has rhodopsin and cones has photopsin. So, okay, so those are the two types of pigments. These pigments will help for the processing of light into neural signals. Okay, so it's a quick recap. In photoreceptor is a cells that can receive light. There are two types. One is rods, another one is cones, and both rods and cones has pigments in it. Rods have rhodopsin, cones have photopsin. Both these pigments are very important thing when the light is transferred or converted into neural signals. So these cells are named based on the shape of these cells. You can see that already these are cones and this has cylindrical shape, so it is known as rods. So what is the basic difference between rods and cones? Rods, they are active in dim illumination. Say for example, when we are going outside in the evening, or when it is about to get dark, or when we are going outside in dark or at night, rods are helping us to see in the dim light as well as at night. Cones, what are the functions of cones? They are active in well lit conditions. Say for example, when we are walking in a daylight, okay, the daytime, okay, the cones are more active, that is helping us to process the light. So rods are helping us to see in the dim illumination, cones are helping us to see in the bright illumination. Okay, so not only that, cones are helping us to see color, okay, as well as cones are helping us for very sharp vision. Okay, rods cannot process color, but rods can help us to identify movements. Okay, in our visual field, if an object is there and if that object is moving, especially when we are driving, okay, when you are driving and if a vehicle is coming from your side, because you will be looking straight and the vehicle is coming from side, okay, so these movement in the side visual field are seen or perceived with the help of rods. So rods have three functions. One, it is helping to see in the dim light. Second one, it is helping for motion movement. Third, it cannot process any light, means any color. Cones is helping us to see in bright light. Okay, it is helping us to see very sharp vision, and also it can process color. Well, now in this slide we will talk more about the different parts of photoreceptor cells. So as we already discussed, photoreceptor cells has three parts. One is outer segment, inner segment, and a connection between outer segment and inner segment, that is known as cilium. Okay, so this is the outer segment and this is the inner segment, and you can see a very small connection between outer segment and inner segment, that is known as the cilium. So here also you can see this is the cilium, outer segment and inner segment. So outer segment is closer, okay, to the retinal pigment epithelium. It contains photopigments which transmits light into neural signals. We already discussed that photopigments, right? Okay, in the previous slide. Okay, two types of pigments, one present in the rods and another one present in the cones. Cilium, as we just now discussed, it is a junction between the inner segment and outer segment, or it is a connecting link. And inner segment, this the whole thing is the inner segment, and that is the location of metabolic activities. So for the cells, okay, for the cells to do this, because photoreceptor cells are the location where the light is converted from light energy is converted into neural signals, so you need a lot of energy, okay, for that processing. So your outer segment is connected to the choroid is supplying nutrients and oxygen, okay, and it is going through the outer segment into the inner segment, okay, where the energy is synthesized within the photoreceptor cells so that light can be comfortably converted into neural signals. So quick recap, it has three parts: outer segment, inner segment, and a connecting link between outer segment and inner segment, that is the cilium. Okay, so now we came into the third layer. So which was the first layer? First layer was retinal pigment epithelium. Second layer was photoreceptor cells, or we also call it as layer of rods and cones. And third one is the external limiting membrane. So we come back to the previous picture, your photoreceptor cells, sorry, your retinal pigment epithelium, and then your photoreceptor cells, and the junction between this is the inner segment and this is the outer segment of photoreceptor cells. So that junction is known as the external limiting membrane. Okay, so it is seen at the base of the inner segments of the photoreceptor cells. Okay, so this is the inner segment and your external limiting membrane is at the base of the inner segments of photoreceptor cells. Okay, so that is the third layer. All right, we are coming to the fourth layer now. Okay, so layer 1 retinal pigment epithelium, layer 2 the photoreceptor cells, and layer 3 the external limiting membrane, and layer 4 is the outer nuclear layer. Okay, remember that this is the outer part or the outer cell, this is the middle cell, or this is the inner cells. So the outer nuclear layer, okay, that means this is the layer, okay, that includes a nucleus of photoreceptor cells. So it is easy to remember the nucleus of photoreceptor cells, okay, is known as the outer nuclear layer. Yes, of course, it contains the cell bodies and nucleuses of rods and cones. So that is the outer nuclear layer. Okay, so here is the next layer that now we will be able to easily tell this is the outer plexiform layer. I already discussed. Plexiform means it is the junction. You can see it over here. Okay, these are the photoreceptor cells and these are the bipolar cells. Okay, so the junction between the photoreceptor cells and bipolar cells are known as outer plexiform. So plexiform means connection. Okay, and so you can see that these are a type of neural cells, these are another type of neural cells. So the junction between the neural cells, right, this junction is known as your plexiform layer. Okay, since it is connecting the outermost photoreceptor cell and the inner uh, ganglion cell, this junction is known as outer plexiform layer. All right, so now we are coming to the sixth layer now. So just a quick review: your retinal pigment epithelium, okay, layers of rods and cones, that is your photoreceptor cells, external limiting membrane, that is a junction between the inner segment and outer segment of photoreceptors. Fourth one is your outer nuclear layer, that is nothing but the nucleus and cell bodies of photoreceptor cells. Fifth one is the outer plexiform, that is junction between the uh, photoreceptors and bipolar cells. And now comes the inner nuclear. Now we are entering into the inner layer, that means the bipolar cell. Okay, the nucleus and cell bodies of bipolar cells forms the inner nuclear layer. Not only that, it also has amacrine cells, horizontal cells, and molar cells. Just try to remember the name of these three cells: amacrine, horizontal, and molar cells. We will not be learning so much in detail about them, but of course there are some functions for this amacrine cell. So you can see that along with the bipolar cell you have amacrine cells, okay, and your interplexiform neurons, their horizontal cells and amacrine cells. You can see that bipolar, horizontal, and amacrine. Okay, you may be wondering what are the functions of horizontal and amacrine and molar cells. It is nothing but it refines the uh, the quality of neural signals, okay, that is being transferred from, means light is transferred to the neural signals, and all these amacrine, horizontal, and molar cell refines or makes the quality of the neural signals better. Okay, so now you will be able to easily remember what will be the next layer, that is the inner plexiform layer. So you remember what is outer plexiform. Now we'll be talking about the inner plexiform in the next slide. Okay, here you go. These are the layer 7, 8, and 9. So it is easy to remember now. The seventh layer is the inner plexiform. It is nothing but the connections between bipolar cells and ganglion cells. Okay, now after the inner plexiform, these are the ganglion cells, and after ganglion cells, these connections of all these ganglion cells are continuing as nerve fiber layer. Okay, so inner plexiform, ganglion cells, and nerve fiber layer. So all these nerve fibers, okay, these will get collected at the optic nerve head and becomes optic nerve. Okay, so that is how each of these layers are functioning. So will be a quick recap about the layers. You can do it by yourself. I do not need to repeat. Starting from the retinal pigment epithelium till the internal limiting membrane. So you need to remember those layers of retina in the order from posterior to anterior. Okay, here comes the last layer, that is the 10th layer, that is the internal limiting layer. It is nothing but the boundary of the retina, and that is sitting closer to the, so your vitreous is here, and the layer between the vitreous and the remaining retinal layers is the internal limiting membrane. So that means that is a layer at which the retina is ending. So you have your choroid, you have the retina, and you have the vitreous. The line or the membrane that is separating the vitreous to the different retinal layers is the internal limiting membrane or internal limiting layer. All right, so now we have completed all the different layers of retina, so it is easy for you also to remember, and if you try to understand the structure in order, okay, from the retinal pigment epithelium to the internal limiting membrane, it will be very easy for you to remember each of those 10 layers in order. And also it is very important to remember those layers in the order. Do not mix the layers, it should be written in the proper order. So make sure that you learn that by writing, repeatedly writing. Okay, so that if whoever asks, you should be able to tell each of the layers of your retina. Now we come into the different regions. Regions is nothing but parts of the retina. Now if you look at this picture, this is how our retina look like when we look through. Suppose if we have a patient, when we look through the pupil of the patient, this is how you see the retina. Okay, you will not be able to see different layers of retina, we can only see the retinal surface. Okay, so this retinal surface is closer to the vitreous, right? So it has two parts. One is the peripheral retina, that means the outer parts of the retina, and you have the central retina. Okay, you can see that the central retina is much more brownish in color. Okay, and it is nothing but because of the pigments of cones. Now you can see what is there in the peripheral retina. Peripheral, this whole thing is peripheral retina. Okay, only this part is the central retina. So peripheral retina, the majority of the retina is retinal periphery. Say this much, these are all retinal periphery, right? And it is designed to detect gross form and motion, as I already mentioned. Okay, your peripheral retina is helping you to see the movement of object. Also gross form is nothing but when you look at a particular point, when you look at your friend's eyes or friend's face, the face is very clear, but the surrounding areas are not very sharp, isn't it? So we are using our central part of the retina for the sharp vision. Whenever we are focusing at some point, the light rays are coming and hitting at the central part of the retina. Okay, but as I mentioned, in our visual field, we can see the other side of the visual field also, but the vision will not be very sharp in the periphery. So your peripheral retina is helping to see for the peripheral vision. Your central retina is helping to see the central vision, or nothing but wherever we are focusing, the central part of the retina is helping us to see clear and sharp. Now you remember we were talking about rods and cones, right? Rods are helping us to see in the dark. So the peripheral retina is filled with rods. Okay, so the majority of the rods are in the peripheral retina. And what is there in the central retina? Okay, as I already mentioned, this is a part for the maximum visual acuity or sharp vision, and this is a small portion. This is the central retina, and they are rich in cone. Okay, so in the central retina we have the maximum number of cones in the central part of the retina. Why? Because that is helping us for sharp vision. Whenever the light focuses into the retina, the cornea and the crystalline lens is focusing the light into the central part. Okay, so you have cones here. Majority of the cells are photoreceptor cells are cones here, and these cone pigments are much more brownish, as well as the retinal pigment epithelium pigmentation is also high. That is why this part is appearing more brown compared to other parts. Okay, and of course there are other parts of the peripheral retina. This is your optic nerve head, and these are the blood vessels coming into the retina. We will discuss about those things in the next slide. So remember that retina has two parts, two regions: peripheral retina and central retina. Peripheral retina has majority or most of the photoreceptor cells in the peripheral retina are rods, and central retina has majorly cones. Okay, so we will discuss about the central retina now. Okay, central retina, as I mentioned, this is the central retina. It has two basic parts. Okay, the whole central retina is known as macula, and the centralmost part of macula is the fovea. There is one more central part of fovea which we'll be discussing later. So this area is the macula, or other name of macula is or macula lutea. Okay, macula lutea or macula, both are same. It is appearing as a darkened region. Okay, so it is easy to remember when you or identify when you look at the retina. The area that is darker, okay, compared to the peripheral area is the macular region. So remember that the bigger area is the macula, and it has more concentration of retinal pigments, and also it absorbs the harmful ultraviolet radiation. So the UV rays that is coming into the eye, it will get absorbed by the macular area. Right, so in this slide we'll discuss a bit more about macula. Okay, so as I discussed you in the previous slide, the macular area is the brownish central area. So we also need to remember its dimension. Okay, it is approximately 5.5 millimeter in diameter, and it is located 3.5 millimeter lateral to the edge of the optic disc. Okay, so this is the optic disc, and this is lateral to the edge. So from the edge of the optic disc to the edge of the macula, that is around 3.5 millimeter, and also it is 1 millimeter inferior to the center of the disc. So when we draw a line from the center of a disc, okay, when we draw a line, okay, your macula, center of the macula is 1 millimeter below the level of the center of optic disc. So why this side is appearing, or why the macular region is appearing very dark? Because the retinal pigment epithelial cells are very tall here. Okay, remember that this is the area where the number of cones are very high, so the retinal pigment epithelial cells are also bigger and taller here. And the retinal pigment epithelium has pigments in it, or melanin pigments in it. That is why the macular area is appearing as brownish in color. Right, so in the previous slide we discussed about macula. You need to remember the macular is a bigger region and fovea is the central portion. Okay, so fovea is the central portion of the macula. Okay, and also it is appearing as a depression. Depression is nothing but it is like a pit at the center of the macula. and its diameter is almost equal to 1.5 millimeter. We will be discussing more about this. Okay, there is one more centermost point of the fovea in the next slide. Okay, here we go. Okay, there is a couple more points we need to discuss about the fovea. As we discussed, the fovea is the central portion of the macula. So the macula is the bigger area, and you can also see that macula is temporal to the optic disc, which we already discussed, and it has the highest concentration of cones. Okay, we already know that cones are helping us to see for the sharp vision as well as to see in the bright light. So this macular region, especially the fovea region, okay, within the macula, fovea is the highest concentration of cones. This many numbers, you can just count it per square millimeter. Okay, that many number of cones are there. And within the fovea there is a capillary free zone. Okay, you know what is capillary? Capillary is nothing but blood vessels. Okay, so there is a zone that does not have blood vessels. Now you remember, here you can see these are the blood vessels on the retinal surface, and all these blood vessels are branching out throughout the retina to give the blood to the retina. You can see all these blood vessels are having micro branches, but in the area of fovea there are no blood vessels. Why there are no blood vessels? So that the light, for the sharpest vision, the light can directly hit and it can go into the photoreceptor cells underneath, because you can see that these blood vessels are on the retinal surface, or you can say that these blood vessels are at the nerve fiber layer level of the retinal surface. That means closer to the vitreous. So when the light hits the fovea, right, or the fovea centralis, there are no blood vessels also, so that the light can easily perfectly go to the photoreceptor cells, which is located in the fovea. So this area that does not have blood vessels is known as foveal avascular zone. Okay, so we know that what is foveal and avascular, that means no blood vessels, and a zone. So there is a zone in the macular region. You can see this will be a bigger macular region. Okay, but at the fovea there are no blood vessels. Okay, these blood vessels are not getting branched into the fovea. So that why the fovea is having no blood vessels, so that the light that is entering can easily go into the photoreceptor cells. Okay, so here we are coming to the central most point of the macula. Okay, so you have a bigger macular structure and you have fovea at the center of the macula, and there is one centermost point of the fovea that is known as phobiola. So it comes like this: macula, fovea, and phobiola. So what is phobiola? It is a central portion of the fovea. It has a diameter of around 0.35 millimeter, and it has the densest population of cones. So you have macula. Of course, the macula has the higher number of cones compared to the peripheral part of the retina. Okay, compared to macular area, foveal area has the more number of cones. Now, within the foveal area, the highest number or the densest area that has the maximum number of cones is the foveola. Okay, so and you can see that this is the foveolar region. It is appearing like a clear depression. That means this is the peripheral retina. It has more retinal layers, but in the foveal region, the number of retinal layers, or foveolar region, the number of retinal layers are lesser. Why? Because the light rays that is hitting here can easily approach the photoreceptor cells. So the number of retinal layers are also less at foveola. That is why it is appearing like a depression. Alright, so we have already completed now the central portion of the retina. We'll discuss about the peripheral retina. In the peripheral retina, these are all the peripheral retina, and before we go into that and blood vessels, we'll discuss about optic disc or optic nerve head. So what is optic disc? Whenever you look at a picture of the retina, you will see a more yellowish and paler color. Okay, paler means it does not have the reddishness like in the other part of the retina. It appears more yellowish. That part is known as the optic disc. Now, what is the importance of optic disc? Now, you need to remember that the light can hit all these retinal surface areas, and the light can pass through all of this retinal surface into the deeper layers, into the photoreceptor cells, means, and from there the light is getting transferred or converted as photons, and the light is transferred as neural signals, and it is coming out. So whatever you are seeing, this orange color here, that is a retinal nerve fiber layer. So all these nerve fibers, okay, will get, okay, from all part of the retina, the nerve fibers are getting collected and into the optic nerve. Okay, so all these nerve fibers are getting collected. It is like a tube, and the nerve fibers are going through this, and which is forming as optic nerve. That is why it is a site where nerve fiber layer accumulate meaning all the nerve fibers coming from the retinal periphery, okay, as well as from the center. It is getting collected through the optic nerve head. So the site where nerve fiber layer accumulate and exit the eye. So it goes out of the eye through the optic nerve head, and behind optic nerve head it will be the optic nerve. Okay, so it has a shape of a vertically oval shape. Okay, it is not a perfect circle, it is an oval shape, and it is vertically oval. Why? The vertical dimension, or this vertical diameter, is more than the horizontal diameter. So what are the things you need to remember in optic disc? One, optic disc is a place where the nerve fiber from peripheral retina, or as well as from the central retina, will get collected and goes out of the eye. So it is known as optic disc or optic nerve head. It has a shape of a vertically oval shape. Why it is vertically oval? Because the vertical dimension is more than the horizontal dimension. Right, a bit more points about optic disc or optic nerve head. And in the previous slide I mentioned it appears paler. Paler is nothing but less reddish or orangish than the surrounding retina. Why it is? Because there are no retinal pigment epithelium behind the optic disc. So optic disc does not have uh, the regular retinal layers like what you see in the other part of the retina. So it does not have retinal pigment epithelium also, that is why it is appearing less brownish. And also there are no photoreceptor cells present in the optic disc, that is why this is also known as a physiological blind spot. Okay, so when the light hits on the optic nerve head, we will not be able to see the light, so we have a blind spot uh, um, in our visual field. Probably when I'm meeting you people after the break, I will be able to demonstrate the blind spot to you people. Okay, and there is one more function for the optic nerve head, and can you see these blood vessels coming out? Okay, so the blood vessel that has to come to the retinal surface is coming through the optic nerve head, and you can see two different types of blood vessels which we will be discussing. That one is the vein, another one is the artery. Okay, we'll discuss about that in the subsequent slide. So optic disc, it is paler than the surrounding retina. Why? Because there are no retinal pigment epithelial cells. Optic disc does not have visual sensation. Why? Because there are no photoreceptor cells in the optic disc, and through the optic disc only the blood vessel, that is the artery, that is coming into the retina, as well as the veins that is going out of the retina. So through the optic disc only these blood vessels are coming in, as well as the veins are going out. Here we go. Okay, in this slide we'll be discussing about the retinal blood supply. Okay, how the retina is getting blood. And we already know that outer retina means the retina that has the retinal pigment, epithelium, and the photoreceptor cell. That is a part of the outer retina. It receives nutrients from the choroid, because just outside the retina we have the choroid to supply the nutrients. The inner retina, okay, that means the retina that is the inner side, that is closely situated to the vitreous or situated next to the vitreous, because vitreous does not have rich blood vessels, so some blood vessels should come to the retinal surface, okay, so that it can supply the retinal areas. So that inner retina is supplied by central retinal artery. Okay, so this central retinal artery enters the retina. It comes to the retina through the optic nerve head, and uh, we cannot see any part of our body that has only arteries. So wherever the arteries are there, there will always be veins, because arteries will bring the oxygenated or pure blood to the site, and the veins will collect the deoxygenated or impure blood out of the site. So wherever arteries are there, there will be veins as well. So the arteries as well as veins are coming or going out of the retinal surface through the optic nerve head. So your central retinal artery, okay, that is the artery that is bringing retina to the retinal surface. It has four branches. It has superiotemporal, superionasal, inferiotemporal, and inferionasal. Okay, we'll understand this a bit more clearer, okay, in the next slide. Right, so this is a picture of the bigger picture of your retina and the retinal, sorry, or the retinal surface. Okay, and this is how you see the retina when you look through the pupil, through the vitreous, okay, onto the retina. Of course, we will not be able to directly see the retina when we look through the pupil. We will have to use additional or specific instruments to see the retina. Now, this is the optic nerve head. This is the macula. Okay, you have your fovea and you have your Phobiola, right? And this macula is located temporal to the optic disc. Okay, so the location of the macula is temporal to the optic disc. Location of optic disc is nasal to the macula. So you will be having our nose here. Okay, suppose this is the right eye. You have nose here. You will have your left eye outside, inside that side. So that is what the positioning of it. So this is the optic disc or optic nerve head. Okay, it is vertically oval because of the vertical dimension is more than the horizontal dimension, and also it does not have any visual sensation because there are no retinal pigment epithelium, as well as there are no photoreceptor cells at the optic disc. So now through the optic disc we have the blood vessels coming in and going out. So when you look at here, can you just see these two blood vessels? One blood vessel is lighter in red, another blood vessel is looking darker and bigger. Okay, you can see this is dark and big, dark and big. This is dark and big. This is lighter, right, and smaller. You can see one darker here, many lighter ones. Okay, so whenever you are seeing blood vessels that is darker red and broader, means bigger, those are veins, and lighter one and thinner ones are arteries. So you can see this is an artery. Okay, this is an artery. This is also an artery, but this is a vein. You can see it is bigger, it is much more darker red in color. So this is a vein, and of course this is an artery, right? This is a vein, this is an artery. Fine. So we can divide four branches of the artery. So now we are talking about the artery first. Okay, so the artery is coming through the optic nerve head and it is going towards the periphery, and this is the superior, right? So we know that macula is located at the temporal side of optic disc. Okay, so that means this is the artery. Okay, superior-temporal artery. Okay, the temporal side at the superior. So this is a superior-temporal artery. You can see this one. And now you can see another one going towards this, that is the infero-temporal. Okay, this or infero-temporal artery. The artery going towards, now this, if this is the temporal side, this will be the nasal side. So you have an artery that is going to the superior nasal, and this is inferior nasal. So this is the superior portion, this is the inferior portion, this is the temporal portion, and this is the nasal portion. So you have superior nasal, superior temporal, inferior nasal, and inferior temporal. So this is applicable to both. Now when I say this is the superior temporal vein, this is a superior temporal artery. This is a superior nasal vein, this is a superior nasal artery. This is an inferior nasal vein, this is an inferior nasal artery. This is an inferior temporal vein, okay, and this is an inferior temporal artery. Okay, so that is how you tell or you specify the name of arteries and veins. And in the next slide, inshallah, we'll be seeing what are the different ways of differentiating between arteries. Right, so we are into our final slide, and I hope it will be a reasonably good experience. Right, so this is into the final slide. Here what we are learning is how to differentiate. So what is the meaning of differentiate? Differentiate means how can you identify, or how can you say one is vein and another one is an artery. So all the blood vessels, whether it is arteries or vein, they are superficial over the nerve fiber layer. Okay, you remember the nerve fiber layer is a ninth layer, okay, which is sitting closer to the vitreous. Okay, that is internal limiting membrane and nerve fiber layer. Nerve fiber layer is the ninth one, okay, sitting very close to the vitreous. Okay, so all the blood vessels are located on the nerve fiber layer. Now veins have larger diameter than arteries. When you look at this, which blood vessel has bigger diameter? Okay, veins are appearing broader and larger. That means the diameter will be of the blood vessels will be more. So veins, the diameter of veins are larger than the diameter of arteries. That is one way of differentiating between arteries and veins. Now arteries are placed over veins. So most of the time arteries will be sitting above the veins. Now you can see it over here. You can see this artery is crossing the vein and it is above. But not always. Most of the time arteries will be placed over veins. Now third differentiating feature is arteries are paler than vein. You can see that veins are darker in color. Okay, that means the blood inside is much more black in veins compared to arteries. So why? Because arteries are having oxygenated blood, pure blood that is having oxygen. That is why it appears light red. And veins are having deoxygenated blood or impure blood. That is why it appears darker. So those are the three differentiating features. Okay, between veins and arteries. One is veins are larger. Second one, arteries are placed over veins most of the time, and arteries are paler than veins, or veins appear darker red compared to arteries. So that completes the presentation of retinal anatomy.